Welcome back. Today we're going to finish our Chapter 5 working cell by looking at metabolism and also by looking at enzymes and what role they play in our cell's metabolism. So if we look at the metabolism or cellular metabolism, basically that refers to all the different chemical reactions that are all occurring inside of a cell. And so a cell's metabolism is going to be made up of literally thousands of different chemical reactions at any given time, again occurring in all those different compartments within the organelles within the cell. And certainly most, if not all, of these metabolic pathways are somehow tied to energy and converting energy, whether it's saving up energy or utilizing energy or changing forms of energy. It's involved in almost every aspect of a cell's metabolism. Now when we think about chemical reactions in general, reactants are the chemicals that go in, products the chemicals that go out, obviously. And one of the common misconceptions is that when we have a chemical reaction, that all the reactant gets turned into product. Because we draw that arrow, it seems like, well, eventually I'm going to have product and no reactant. But that's not the case in chemistry, but instead chemical reactions will proceed until they reach what's called the equilibrium. And equilibrium means there's no net change in the reactants and products. And what that equilibrium exactly is depends on the chemical reaction and the environment it's in. And so, for example, maybe 90% of the reactants are going to get turned into products, but 10% are going to remain reactants. And remember, chemicals are in constant motion, and so eventually the reactants may be still breaking down into products, but those products may be bouncing into each other and reforming into the reactant. And so when we reach that equilibrium, we're not going to see the reactant continue to disappear. Okay? And so we're not going to see that 100% completion like we might expect intuitively when we look at a chemical reaction. And again, that all has to do with chemicals being in constant motion. So keep that in mind, that chemical reactions don't proceed to 100% completion, but they're going to proceed to that equilibrium point. And chemists divide chemical reactions into two main categories dealing with energy issues. And again, obviously in living organisms, that's going to touch home with us as well. And in particular, an endergonic reaction is a chemical reaction that requires energy to be added. And so this is a reaction that cannot occur unless we have an outside energy source that's going to be added to that. And that's usually going to involve building larger chemicals. And so when we build proteins, for example, we require energy to be able to do that. And if we add energy as we're building a protein, that energy, at least some of it, is going to be stored up as potential energy. And so we think of endergonic reactions, energy has to enter, so think enter and ender, and some of that energy is going to be stored up in these large macromolecules that we're building. Okay? So for example, in photosynthesis, plants take low energy small chemicals like carbon dioxide and water. They're going to build a high energy chemical that's larger, like glucose, our sugar there, their C6H12O6. Now that glucose has lots of potential energy stored up. That energy had to come from somewhere. Obviously in photosynthesis, that energy comes from the sun. That's the input that's driving that reaction from the product side to the reactant side. Okay? And if we look at that in a graphical form, in an endergonic reaction, the reactants are low energy, again, like carbon dioxide and water. The products are high energy, like glucose. And so to get from reactant to product, we have to add a certain amount of energy. And again, that makes it endergonic. The opposite of that endergonic reaction then would be an exergonic reaction. And exergonic reactions are going to release energy. So think exergonic exit. Energy exits these reactions. And since storing up energy involves building chemicals, releasing energy is going to involve breaking chemicals down. So exergonic reactions, our reactants usually are going to be large chemicals. Like in this case, when we take glucose in our cell metabolism, we're going to break it down to release energy. We're going to take that high energy glucose and break it down into low energy products, like carbon dioxide that we breathe out. Okay? And again, graphically, if we take a look at that, our reactant high energy, our products low energy, and so when we move from reactant to product, we're releasing a big chunk of energy, and that would make it an exergonic chemical reaction. The most important chemical in your cells regarding energy and this whole issue of metabolism and exergonic and endergonic is a chemical called ATP. And ATP is just an acronym for adenosine triphosphate. ATP is a very small chemical in your cell, and it represents your cell's energy currency. For example, it represents cash. If we use an analogy, ATP is cash, is money ready to spend, versus energy that might be stored up in a sugar or fat. That energy is present, but the cell can't get to it right away. That might, for example, refer to money you have in a savings account somewhere. You have it, but you can't get to it right now. Versus ATP, that represents energy that's in a usable form. It represents cash. It's ready to spend right now. And almost everything in a cell that happens that needs energy needs ATP. ATP really is a central important chemical to your cell, and in fact cells can't survive more than 2 or 3 minutes without ATP. So if your cell cannot generate new ATP, that cell dies very quickly because so much in the cell depends on that ATP for energy. Okay? And that energy associated with ATP is going to be used to couple or link different chemical reactions together. We don't see chemical reactions occurring just isolated, just here's one chemical reaction in cells, but instead chemical reactions are usually linked or coupled together into long chains or long chemical pathways, and ATP is often involved in that coupling or linking. The reason that ATP can be such an important chemical is it has an interesting chemical bond called a phosphate bond. And a phosphate bond contains a lot of energy, and so when that phosphate chemical bond is broken, it releases a large amount of energy very quickly. And so let's take a look here. Here's ATP on the left. It's a nucleic acid, so that's where it falls in terms of our macromolecules. But notice it's got 3 phosphate groups, the triphosphate. Well, each phosphate group has a very strong negative or minus chemical charge to it. And we know that if I put 2 minus things together, like 2 phosphates, the natural reaction is for those two things to repel each other, because those like charges tend to repel. So we're going to take with a phosphate bond 2 things that tend to repel each other and hold them close together. And so it takes a lot of energy to do that. And that's what the little pink squiggly lines represent between those phosphates, those high energy bonds. Then when your cell needs energy, it can break one of those phosphate bonds. It's going to release a large chunk of energy very quickly in just one easy chemical step. And again, ATP is critical for everything in a cell. It's ATP energy that allows a lot of these endergonic reactions to occur. For example, when a cell needs an endergonic reaction that requires energy, that energy usually is going to come from ATP. When your muscles move back and forth and you contract your muscles, that's ATP that's releasing the energy for those muscle fibers to contract and move along. And it's ATP for cell transport. When we talked about active transport last time, we said it required energy to work that sodium-potassium pump. Well, that energy comes from ATP. A phosphate is broken off, that energy is released, and that's going to pump those sodium and potassium ions across membranes. So let's take a look at ATP and this idea of reaction coupling. And again, reactions are coupled or linked together here. And so ATP is kind of recycled. That if you've got your ATP, it's high energy. When your cell needs energy, it's going to break a phosphate off and release energy. And certainly breaking that phosphate off represents an exergonic reaction. It's releasing energy. Likewise then, if your cell uses a lot of ATPs up, now it has a lot of ADP, which stands for diphosphate, and phosphate groups floating around. And so your cell's going to recycle those back into ATP. To do that, your cell has to add energy. Okay? And so that would be an endergonic reaction to build ATP, an exergonic reaction when your cell tears it apart. Okay? And so as these chemical reactions are coupled, ATP is going to kind of be the main coupling ingredient, if you will. And so, for example, if you have an endergonic reaction that has to occur, it needs energy, your cell will break down ATP, that energy will then power that endergonic reaction. Likewise, if you have an exergonic reaction occurring that releases energy, your cell may take that energy and store it up as ATP, use it to connect ATP together. Okay? And so again, these exergonic and endergonic reactions are always coupled together. Energy being released is going to be used in the next reaction. If energy needs to be stored up, it has to come from a source in those endergonic reactions. In addition to ATP and energy, another really important factor about your cell's metabolism are enzymes. And enzymes are proteins that are going to speed up chemical reactions. And all chemical reactions in living things, it doesn't matter whether it's endergonic or exergonic, it doesn't matter what living cell we're looking at, all chemical reactions require enzymes. Enzymes can digest your food, they can actually build chemicals. And again, any pathway, every reaction is going to require its own enzymes. Now enzymes are proteins, so they're long chains of amino acids, and they're going to act as catalysts, meaning they're going to speed up chemical reactions. They're going to make your metabolism happen much faster than it would in a test tube or in a beaker somewhere. In your cell, it occurs much quicker because those enzymes are present. Okay? Enzymes often have an ASE or ACE suffix, so anytime you hear something like sucrase or lactase, you know you're dealing with an enzyme. And the way enzymes are going to speed up chemical reactions is they lower what's called the activation energy. Okay? So enzymes function by lowering activation energy. So I want to take a look at what this activation energy is, and then more importantly, how are enzymes going to affect or lower that energy. The activation energy of a chemical reaction is the amount of chemical reaction that the reactants have to absorb to get the reaction started. And all chemical reactions require some activation energy, some amount of energy to get started. Some a lot, some a little, but they all require that activation energy. And it doesn't matter whether it's endergonic or exergonic. This energy has to be present. This activation energy is needed. And one way to kind of think of this activation energy is to think of it as a barrier. For example, your body is made up of millions of molecules of chemicals, lots of sugar, lots of glucose, for example. Well, how come all your glucose doesn't just suddenly burst and break down? Okay, you kind of spontaneously combust, if you will. The reason for that is this activation energy, that for glucose to start breaking down, all those glucoses would have to absorb some energy. And that means your body would have to absorb an awful lot of energy and apply it directly to the glucose to get that to happen, and that's certainly not going to happen. And so all cells have to have some sort of activation energy to add to reactants to make those reactions happen. And the way enzymes are going to work to speed up chemical reactions is they're going to actually lower the amount of energy needed to get started. They're going to lower that activation energy. And so here we see a graph. The black line represents the normal reaction with no enzymes. And so to get from reactant to product, this would represent an exergonic reaction because it's going to release some energy. But even an exergonic reaction needs that activation energy, represented with an EA on the chart there, to get started. And we can think of that again as a barrier or sort of a hill that has to be climbed. Now if we add enzyme, that's what it's showing with the red line. In the enzyme, notice the energy in the reactants and the products are the same. So we're not really changing the chemical reaction, but it takes less energy to get started. Our activation energy hill is now just a speed bump, okay? Which means since reactants need less energy to get started, they're going to move to products much more quickly because we're not going to have to find extra energy to add like we would if there was no enzyme present. And that certainly begs the question, well, how do enzymes work? How can an enzyme kind of magically, if you will, lower that activation energy or how much energy it needs to get started? And enzymes do this in a number of different ways. First of all, they can bind the two reactants and hold them close together. That's going to foster the chemical reaction. Not only that, but they're going to hold the reactants in the right orientation. So they're going to put the reactants in the right place, again, so that a reaction is going to be likely to occur. In addition, these proteins that make up enzymes also create little microclimates or microenvironments, depending on which amino acids are present in that particular enzyme. And so it could create a very small pocket that's really acidic or a very hydrophilic pocket that may foster a chemical reaction. So it's kind of a combination of getting the reactants together correctly, but also creating these little microenvironments that are going to increase the likelihood of the reaction occurring and going from reactant to product. And here we can see we talk about enzymes through a model called the lock and key, the idea that enzymes have a specific shape just like all proteins, and the most important part of the shape is called the active site. And that represents sort of the lock. That's the part where the reactant is going to bind to it. And so the reactant is going to bind to that active site and actually fit or lock into that active site. And so the reactant represents the key, if you will. And since enzymes are so critical, I want to take a few moments and talk a little bit about enzyme structure and characteristics, and particularly how they're going to be worked and controlled in your cells. And so, again, they are proteins, so they are these chains of amino acids. And remember, we make our proteins through a polymer called a polypeptide, which is just usually hundreds of amino acids hooked together. They're going to fold together into their correct shape, and then the protein may be one or more of these polypeptides then that all have to physically lock together. And that the most important part of a protein is the sequence of the amino acids, which amino acids we put together, because that's going to directly determine shape and function of any protein. Okay? And within the enzymes, I mentioned that active site, that is the part of the enzyme that's actually going to bind to the reactant. So it's going to have a very specific chemical properties to it and a very specific three-dimensional shape, just depending on which amino acids make up that active site. And so if we look at an example, here's a chemical reaction. H2O2, which is hydrogen peroxide, is a common toxin in your body. It's a common metabolic product. Your body has to be able to get rid of it, otherwise it can be toxic. And so we break peroxide into two harmless things, water and oxygen. Okay? And so obviously in this case, peroxide is the reactant and water and oxygen are the products in this chemical reaction. Well, we have an enzyme to make this occur, because if we had to wait for peroxide to break down on its own, your cell would be dead. It would become too toxic. Your cell has to make it go away much quicker. And so we have an enzyme called catalase, and catalase will break down peroxide into water and oxygen very, very fast. Okay? Now, we call peroxide a reactant normally, but if an enzyme is present, we're going to call peroxide a substrate. And so substrate is still a chemical reactant, but by calling it substrate, that tells us there's an enzyme present. And so when you see that term in your book, substrate, it's just the reactant in an enzyme sped up chemical reaction. And so catalase is going to bind peroxide in its active site and convert it into water and oxygen in this case. Remember, in all of our topics, structure always relates to function. How something is built always determines what it can do. And so an enzyme's function and characteristics are going to be directly related to that idea that they are proteins and have their specific shapes. Okay? And one issue with enzymes is they are very selective, meaning the substrate has to fit into that active site to speed up the chemical reaction. And again, your book has models where shape is important, but remember, chemistry is also important. It's just hard to draw on a kind of easy picture there. So, for example, catalase will speed up the reaction of breaking down peroxide into water and oxygen, but that's the only chemical reaction catalase will speed up. So if your cell has 1,000 chemical reactions going on, you need 1,000 different enzymes to speed up every one of those different reactions. Enzymes can be recycled or reused, and so the enzyme is not a reactant, it's not a product. It's more like a bystander. And so once one catalase binds peroxide and breaks it down, it's going to release the reactants. Now that catalase can bind another peroxide and break it down, and then bind another. And so a single enzyme can be reused over and over again for that same chemical reaction. Enzymes are not going to change the basic chemistry of a reactant. So remember, we said those chemical reactions go to their equilibrium point. If I add an enzyme, it's not going to change the equilibrium point. Now the reactant is going to get there a lot faster. It may reach equilibrium in a matter of seconds or minutes instead of hours or days, but it's not going to change the basic chemistry. And along that same line, the last thing I want to mention with characteristic here is that while enzymes speed up chemical reactions, they cannot make new chemical reactions occur. Meaning, if two chemicals normally don't bind together, we can't add an enzyme and magically make those chemicals stick to each other or bind together. So all the reactions in our body would normally occur, but enzymes are going to make these reactions occur much faster. And again, if they didn't, we couldn't sustain life. If we had to wait for food to break down on its own, by the time it did and released its energy, our cells wouldn't be alive anymore. And so we need these enzymes to speed up metabolism to maintain life. Let's look at a drawing here of an enzyme and how it works. And so number one, there is a typical enzyme. In this case, it's meant to represent sucrase. Again, think of that ase suffix there, the active site with a specific three-dimensional shape in chemistry. Our substrate or reactant, in this case, is sucrose, a sugar. It's going to bind to that active site. Now, that binding to the active site is only temporary, and it's a very instantaneous thing. As soon as it binds, the products are going to be formed. The enzyme will spit the products out, in this case, our two monomers, glucose and fructose. Okay? And now the enzyme's ready to be used again, and so it can bind the next sucrose, spit out the products, and so on. And so that enzyme is going to be used over and over again. If we take a look then at an animation, and these animations, by the way, are all coming from the mybiology.com website. You can go look at them anytime you'd like to with their voiceover associated with all these different things. But here we see our reactant getting turned into product. And again, this is an exergonic reaction. It's going to involve the release of energy. But even exergonic reactions require that activation energy. So those reactants have to absorb a large chunk of energy to get started, to get over the hill, to get turned into product. With an enzyme present, things are going to happen much faster. And so an enzyme is going to have a three-dimensional active site. That active site is going to be the right chemical shape for those reactants to fit into it. Not only that, but it's going to create a microenvironment, a small chemical environment, either acidic or basic or hydrophilic or hydrophobic, that's going to allow those reactants to get turned into product much faster. And so the reactants, we'll now call them substrates with an enzyme present, will bind to that enzyme. The activation energy now is going to be much, much lower. It's going to take a lot less energy to get started. Those reactions are going to get turned into products. Once those products are released, now our enzyme looks the same at the end as it did at the beginning. Again, we're not using up our enzyme. It can bind to the reactants a second time and be reused over and over again. And so this is a pretty nice animation showing this. The one shortcoming of our animation is, remember, this is happening quickly, and this animation is agonizingly slow, but this enzyme would be working very quickly, speeding up a reaction over and over again. If we think about speed, how fast do enzymes work? They go from anywhere from 1,000 times to 1,000,000 times faster than the reaction would occur by itself with no enzyme. So again, they're substantially speeding up all these chemical reactions. And while technically a reaction occurs whether there's an enzyme or not, again, the reaction with no enzyme occurs so slowly we can hardly even measure it. For example, peroxide breaking down into water and oxygen is going to reach its equilibrium under normal conditions in a beaker somewhere, maybe over the weekend, right, a couple days, versus if I add that enzyme catalase, it's going to reach its equilibrium in less than a minute, and so very, very fast. And so if you're missing one of your enzymes, basically somewhere in your metabolic pathway, there's one reaction that is not occurring. It's occurring so slowly. Okay? And again, because each enzyme is selective, each enzyme only speeds up one chemical reaction. Each of your cells need thousands of different chemical reactions. And if metabolically you're missing just one enzyme, that may result in very serious health issues. And a good example of that is a relatively rare genetic disorder called ADA, which stands for adenosine deaminase deficiency. And there's that ACE suffix. And so adenosine deaminase is just one of these thousands of enzymes that we have. In particular, adenosine deaminase breaks down chemicals, proteins, into urea, which then you can get rid of in your uric acid. And so if you have ADA, you're missing this enzyme, you can't break it down, these chemicals, these protein byproducts become toxic, and certainly can cause serious disabilities. In particular, the main symptom of ADA is your immune system breaks down because these toxins build up in your white blood cells and start killing off your white blood cells, and the immune system no longer works. Okay? And so again, just missing one enzyme can be have a drastic effect on health. Another important aspect about enzymes is the control of enzymes. Enzymes are proteins, and proteins are very expensive, if you will, to make for your cell. They require lots of energy, lots of amino acids, and so it's not an easy thing for a cell to make a protein. And so if your cell has an enzyme that's speeding up the chemical reaction, let's say the cell has enough of the product, it doesn't need the reaction to occur. Cells simply don't tear apart or break apart the enzyme and then have to rebuild it a half hour later if it needs a reaction to occur again. And so cells can't simply break down and rebuild enzymes to turn them on or off. It's just too expensive. And so instead, cells use a group of chemicals called inhibitors to help regulate or control enzymes. And so an inhibitor is any chemical that's going to interfere with an enzyme's activity. Again, slow down that enzyme. Basically, it's an off switch. That inhibitor will bind to an enzyme and turn it off. Now, remember, chemicals are in constant motion, so this is not a permanent off switch. That as that inhibitor and enzyme bounce around, eventually they're going to bounce apart. That's going to turn the enzyme back on. And so if the cell needs the enzyme to stay off, it has to keep producing inhibitor to keep binding to and keeping turning off that enzyme. And there's a couple different ways inhibitors work. We see some inhibitors are competitive and others are referred to as non-competitive. So let's compare and contrast those two things. A competitive inhibitor is an inhibitor that's going to bind to the active site. And so it's physically going to lock onto that three-dimensional active site of the enzyme. And once it does that, it's just going to block the substrate. It's going to physically be in the way. So now the substrate cannot get to the enzyme, so the enzyme is effectively turned off. Now remember, we said that active site had a very specific three-dimensional shape in chemistry to bind to the substrate. So if the inhibitor binds in the same place, that must mean the inhibitor chemically must be at least moderately similar to the substrate to fit and to chemically bind into the same place. The other type of inhibitor is a non-competitive inhibitor. Non-competitive inhibitors will bind basically anywhere else on the enzyme except for the active site. And so if it binds someplace else, what that does then is causes the protein or the enzyme to change shape when the inhibitor binds to it, and so the active site is still there, but it's not going to be the right shape, so the substrate is no longer going to fit into it. And this can be any chemical that binds to the enzyme. It doesn't have to be similar to the substrate because it's not binding to the active site. So if we take a look, here's a typical enzyme substrate model. A competitive inhibitor is going to bind to that active site like we see on the left there and just physically block or get in the way of the substrate. A non-competitive inhibitor binds someplace else, but when it does, it changes the shape of that active site, so now the substrate no longer fits in. Okay? So they go about it differently, but the end result is the same. The enzyme is temporarily turned off. Once the inhibitor breaks free of the enzyme, then the enzyme will be turned back on. And a common way that these inhibitors work is through a process called feedback inhibition. And as we mentioned earlier, chemical reactions don't occur isolated, but they are in chains. That reactants get turned into product, that product then is a reactant and gets turned into the next product, and so on down these long metabolic pathways. And oftentimes it takes many small steps or many minor chemical reactions all together to produce a particular product. And so let's take a look at that and this idea then of how an inhibitor might control that pathway. Let's say chemical A is a nutrient. It's something that the cell takes in. It's kind of our starting block. And this chemical G is a chemical that the cell needs. The cell uses up this chemical, so it has to constantly produce G and replace the G that it's using up. Well, to do this, it'd be nice if A could just be converted into G in one big chemical step. But again, typically that's not what we see. Instead, we see lots of small chemical reactions. And so A is going to get converted into chemical B. To do that, A is the reactant and B is the product, and we need an enzyme. We'll just call it enzyme 1 to speed up that chemical reaction. Once B is produced, that B now is going to serve as a reactant, and it's going to get converted into another chemical, let's say chemical C. Okay? To do that requires a second enzyme. And it's going to go right down the chain. Another enzyme maybe converts C to D, D to E, and so on. So that to produce this chemical G requires several different chemical reactions and requires lots of enzymes. And again, this is why we need so many enzymes in our system. Well, let's say that this cell makes all these enzymes, you have plenty of A, the metabolic pathway is turned on, and you're producing lots of G, and now the cell has enough of this product G. It doesn't need any more. Okay? Well, what we see is oftentimes in this feedback inhibition is one of the products near the end of these metabolic chains, in this case we'll just pick on G, is not only going to serve as a product for the pathway, but it's also going to be an inhibitor to one of the early enzymes. And in this case, if there's a lot of G around, it will be a non-competitive inhibitor and bind to enzyme 1, which will effectively shut down enzyme 1. Okay? And so in a pathway like this, we don't need, if we've got 6 enzymes, we don't need 6 inhibitors. We just need 1, because if we turn enzyme 1 off, as soon as all the A is used up, we won't make any more B. And again, that'll kind of eventually shut down the process. And so this feedback inhibition, what it does is creates these kind of self-regulating loops. For example, when there's no G present, there's no inhibitors, the pathway gets turned on and your cell starts making lots of product G. If there's too much G, it starts to bind to enzyme 1 and it turns it off. Now as G gets used up, you have less and less G. When it breaks free from the enzymes get turned back on and you start making more G. And so that's feedback inhibition, and that's the most common way our long metabolic pathways are regulated. Many poisons that are detrimental to organisms are poisonous because they inhibit enzymes that normally don't get inhibited, or they tend to bind to the enzyme so strongly that it is more of a permanent off switch instead of a temporary off switch. For example, a lot of antibiotics, penicillin, our first antibiotic discovered, is a poison for bacteria cells. It turns out that penicillin binds to an enzyme and turns an enzyme off that bacteria need to make their cell wall. And so when you take penicillin, the bacteria cannot produce their cell wall, so they can't reproduce, and eventually they're going to die off because they can't finish their cell division. A lot of pesticides, you go to Home Depot and buy some wasp killer to kill a hornet's nest, for example. The reason that those insecticides are toxic is because they're inhibitors to certain neurotransmitters that normally those enzymes don't get turned off. And so they bind to the enzymes and they don't allow their nervous system to reset. And so once a nerve fires after that spray, it won't fire again. And eventually, because of that paralysis, the organism dies. Now we mentioned that enzymes speed up chemical reactions, but exactly how fast a reaction occurs is going to not only depend on enzymes, but lots of other factors as well. And there are many different factors that may influence the efficiency of how well an enzyme works. And we'll be doing a lab looking at enzymes, in particular, some of the factors that may influence these issues, and again, we'll look at these in lab. The enzyme concentration, so how much enzyme we have, certainly is going to influence how fast a reaction might go. The substrate concentration, how much reactant are we starting with? Okay. Temperature certainly may influence that, or the pH, the kind of chemical conditions. And so, just as an example, I want to look at temperature and how it may influence reaction rate. Remember, temperature is just energy, and increasing temperature means we're increasing energy. And so it makes sense to think about the idea that if we increase temperature, we ought to increase the rate. Things ought to happen faster with more energy. And what we see is as we gradually increase temperature, we will see that the rate goes up and up and up. The reaction is going to occur faster and faster. And when it's occurring at its absolute fastest, we call that the optimum temperature. That's the temperature where the reaction is going to occur as fast as it possibly can. But if we continue to increase temperature above the optimum, what we see is, surprisingly, the reaction rate will plummet, and it will decline very fast, and basically the reaction will stop occurring. Okay? So if we take a look at that graphically, here's temperature on the x-axis and reaction rate on the y. Again, at that optimum, we're going to have the fastest reaction rate. But if we go above that, the enzyme is going to denature. And denaturing of an enzyme means we're going to change the enzyme's three-dimensional shape. So eventually, we're going to add so much energy into it if we keep increasing the temperature, basically we're going to cook the enzyme. We're going to break it. And once we break down that enzyme's three-dimensional shape, its active site is no longer going to bind to the substrate, so that enzyme is going to no longer function. And certainly temperature can denature or break apart an enzyme, or chemicals can do it. Things like pH or other chemical conditions might act to alter that enzyme's three-dimensional shape. And again, we'll look at temperature and several other factors and their effects on enzymes in lab. Wrapping things up, we went over cell metabolism. We compared those exergonic and endergonic reactions, and we looked at ATP. Not only the importance of ATP as an energy source, but also as that chemical that's going to allow your reactions to be coupled or linked together. And then we spent most of our time talking about enzymes. Remember that activation energy, the energy to get the reaction started. Enzymes are going to lower that activation energy. Enzymes are proteins, so they're chains of amino acids. They have that very specific three-dimensional active site, and shape is going to be very important for those proteins. We talked about the function, the idea that enzymes are very specific or selective, and that they can be reused, and that they're not altering the basic chemistry of the reaction. They're just making things happen much faster. And then we talked about some inhibitors, competitive and non-competitive inhibitors. Also, we looked at feedback inhibition as a control mechanism. And then we finally finished up looking at various factors that may influence how efficiently enzymes work or the rate at which a reaction may occur.